Hi, and today we're looking at inheritance in action. This is a great video for GCSE, but also if you're studying A-level and you're fairly new to the topic and you want to make sure you've got your facts straight, start here. What we're going to be looking at is the following things. We're looking at the fact that different forms of a gene are called alleles and that they can be described as dominant or recessive. So even if you haven't studied this, most students have got a fairly good idea of what that means. So to put it in context, you might have the gene for a characteristic such as eye colour, and there's two forms of the allele, brown eyes or blue eyes. There may be a gene for petal colour in roses, and there may be an allele for red and an allele for white. So an allele is a form of a gene. A gene codes for a characteristic, an allele gives a variance of that characteristic. We're going to predict genetic crosses and we're going to work backwards from the genetic cross to work out what the genotype of the parents were likely to be, and we'll work out how to work out those Punnett squares and set your work out so you get the full marks. You can have a look at this in your own time, and there are several definitions that fit. The kind of one I would hang on to things like a recessive allele. And a recessive allele is an allele that's only expressed if there isn't a dominant allele present. In other words, the dominant one will be expressed over the recessive. So it's almost like a weaker gene, a weaker allele, but you wouldn't write that. You're just going to write a recessive allele is one that is not expressed if there's a dominant one present. Now, if that doesn't make sense, it will as we move through the video. Here's a word, I'll just draw attention to it, diploid. That means we have two chromosomes. You have one from mum and one from dad. So your chromosomes come in pairs. We actually call them homologous pairs, and most organisms operate that way. You get one from mum, one from dad, boom, and they're your chromosomes. However, there are some organisms like non-flowering plants where they are monoploids and they just operate using one chromosome. And if you think about, say, for instance, bacteria, they use one chromosome, one circular piece of chromosome. So not all organisms are diploid, but certainly we are and higher organisms are. When I talk about higher organisms, I mean more complex ones with circulatory systems and so on. So this bit is new, and it's new because it links to the previous lesson where we looked at mutations. Now, I've just had a lesson with my year 11 and we were talking about how do mutations happen. And one of the things I said to them was, well, if you copied out everything from that textbook, would you make mistakes? And of course the answer is yes. What kind of mistakes would you make? Well, you might write some words twice. You might miss out a word, or you might misspell a word. And these are all exact interpretations of what happens with a mutation in your cell. Every time a cell is copied, every time a cell divides, it needs to copy its DNA. And the sequence, the nucleotide base sequence, can be copied wrong, it can be missed out, or it can be duplicated. And all these things lead to what's called a mutation. So it's a really good analogy, the idea of copying out the textbook. You're going to make mistakes. When your cell is copied, mistakes can happen. Now they happen, as we said last lesson, more often than we would think. So why don't we have more problems? Well, remember we said that a lot of the time the mutations happen in non-coding parts of DNA. That's right. We said that not all your DNA codes for proteins. There's some of it where we're still trying to figure out what it actually does. And if you end up with a mutation in that type of DNA, it's not such a big deal. Also, we said that some mutations can be beneficial, or the enzyme, say it alters the amino acid sequence and the active site is changed. Maybe it's not changed significantly enough that the enzyme won't work. So we don't notice the many mutations that take place as often as they do. Now we're going to look at this example of a plant. We're looking at the petal colour of the flower that the plant produces. And it says a ruby red is the local common name given to a garden plant that produces a red petal flower. And it's an enzyme that's responsible for making the red pigment. So we've got this molecule that we're just going to call a pre-pigment. And here's the enzyme that changes that into the red pigment that gives the petal that colour. So if we have a mutation in the base sequence, we get a different amino acid, or certainly we get a different sequence of amino acid, which means that the polypeptide is going to fold differently, which means that the enzyme will have a different active site, which means it can't actually work. So a mutation occurs in the nucleotide base sequence that codes for an amino acid in the enzyme responsible for the production of this pigment. So the pre-pigment is not changed into a pigment because the enzyme is mutated. And that's what results in our recessive allele for the gene petal colour. So let's have a look at the phenotype that would be expressed from different genotypes. So if we've got big R, we're going to get the phenotype, the petals will be red because enough pigment is being made. If we have the genotype big R, little r, we still have the enzyme. And so the red colour is still made, so the petals still appear red. Only if both of the codes, both of the alleles for the enzyme are recessive, only if both of those codes are missing, would we have a white petal. So how would you set your work out with that in mind? Let's consider we're crossing a homozygous red with a homozygous white. So let's have a look where we are with that. We already know how to represent our alleles. We used big R for the colour red, and we used the small r for the white colour, the lack of pigment. So we've written down our phenotype. We were told we've got homozygous red, which means both are the same, homozygous, and we've got a homozygous white. In fact, to get any white, they have to be homozygous because it's recessive, and we're going to cross those. So here we've got our genotypes, and here we've got the possible gametes that we can produce. We can only produce a gamete with a big R, with a dominant allele from this plant, and we can only produce a gamete with a recessive allele from this plant. So what we do is we circle them to show that they are gametes, that they are discrete in nature. So from our cross, we can see that the only possible combination we can get is a big R, little r, which is heterozygous. This is dominant over this, so therefore all our population are going to be red in colour. Now, sometimes, because this is the first cross we've done, we call all of these our F1 generation. And sometimes questions will ask, if you then cross the F1 generation, what are the proportions, what are the ratios of offspring that you will get then? So let's have a look at that. Okay, so my phenotype was red, and it was times another crossed with another red plant. But we knew that the genotype had a dominant and a recessive allele, so we know that that was indeed the cross, and we know that the gametes produced, well, this will go into this one, this can give one of those, that will give this one, and that one will give that one. And then when we cross them, just working roughly, clearly if you're doing your working in your book, or if you're in an exam, you try and make sure you lay your work out nice and neat. And also notice how we've chosen uppercase letter that is very different in style from the lowercase letter. You wouldn't choose a letter that was O and O like that, because you're going to get mixed up as you do your diagram. So we need to put our gametes into our Punnett square, and we're going to do big R, little r, big R, little r, circle them to show that they are indeed gametes, and then as we cross them. So we're going to have, this is going to be red, this is going to be red, this is going to be red, and this one lacks the red pigment, so it's going to appear white. So our phenotypic ratio, what's expressed, will be red to white in the ratio 3 to to 1. And it's useful to remember what produced that 3 to 1 ratio. You had a heterozygous and you had a heterozygous. You got a 3 to 1 ratio. Now remember, it's not an exact science. Just like if you've got 4 children in your family, you don't have 2 boys and 2 girls. The chances are that you would have 2 boys and 2 girls, but in the real world, things occur more randomly, and so it's only a suggested 3 to 1 ratio. But how this might appear in a question would go like this. It might tell you that you have a scientist was investigating pea plants, and there were yellow and there were green pea plants, and what he noticed was that when he took a green pea plant and he crossed it with another green pea plant, what he noticed was when he used 1,000 seeds was that 757 turned out to be green, but what he noticed was that 243 turned out to be yellow. And the question then might ask you, what is the likely genotypes of these 2 green plants? Well, let's think about it. We are assuming that green is dominant to the yellow colour. And we are noticing a 3 to 1 ratio, roughly. Now from our previous cross, we noticed that a heterozygous times with a heterozygous gave us a 3 to 1 ratio. So let's just check that out. Let's remove this, and what we're going to say is we're going to say let big G equal green, and we're going to say let little g equal yellow. And let's work out that cross. We're going to say that the phenotype would be big G, little g. How do we know that? We're guessing that from our cross. And we're going to be timing, phenotype is what's expressed, how it looks. So it would be green times a green. Genotype, that's what alleles it has. So it would be big G, little g, times big G, little g. Why? Because when we got our information from the question, it told us that there was this ratio, and we're recognising that that is roughly a 3 to 1 ratio. So we're now going backwards to check. The gametes that can be produced from that are big G, little g, big G, little g. Let's stick them into our Punnett square and check it out. So over here, I'm just going to quickly work it out. Big G, little g, big G, little g. And here we get a green plant. A green plant, it's genetically different, but phenotypically the same. A green plant and a yellow plant. 1, 2, 3 green to 1 yellow. It fits with the same ratio, and so therefore working backwards on our expected offspring, we can then work out what our anticipated genotype was for the parents. So these would be described as your F1 generation, and it would ask you what is the expected genotypes of the parents, and you would work backwards. We're going to do more examples. Let's stick with the same example and imagine that we've got green pea plants and yellow pea plants, and that green is dominant to yellow, and the green is represented by a big G and the yellow is represented by a little g. I didn't choose to use yellow and yellow for green and yellow because they are lowercase and the uppercase are too similar and I could get mixed up in my diagram. So if a question tells me that I have a homozygous green and a homozygous yellow, what would be the expected ratio. Well, I've got green and I'm timesing it with a yellow. The genotype, if it's homozygous, both of the alleles are the same. Big G times little g. This has to be homozygous for it to be yellow. And the gametes, therefore, can only be... So now if I work that out on my Punnett square, I've got big G, little g. I always put the uppercase letter first. It's just etiquette. It wouldn't be wrong if you did it the other way at GCSE. It's just etiquette. So what I would find is that if I had a pure breed and a pure breed, all of my F1 generation would be green. So therefore, in a question, if you've got a plant, for example, and go back to the red, the ruby red flower, and it tells you that it takes the ruby red and it crosses it with a white flower, and it tells you that 100% of the offspring are red, or have red petals, and it asks you what are the likely genotype of the parents, well, you would know that when you cross a pure breed with a pure breed, then you would know that this is actually a homozygous and not a heterozygous. So you would know that this was actually big R, and this was little r. So the point I'm trying to get to is that we can use phenotypes and genotypes to work forward and predict the phenotypes and the genotypes of the offspring. But looking at the ratio of offspring produced, we can work backwards and we can find out what the genotypes of the parents were. How? Because if we have 100% of one colour and we've crossed a recessive with a dominant, that dominant, the genotype, must have been a pure breed. If we've got one where, again, we've got a red flower and we cross it with a white flower, we would then expect a 3-to-1 ratio. So working from those ratios and working backwards, we can work out what our genotypes are.